All right, good morning, everybody. Let me make sure that everything is working out correctly because I was having some issues with my stream earlier today. Getting everything set up. Oh, I need to shut this light off, then turn this light on. Camera looks okay. All right, so today one of the things that I wanted to do is continue on from last time where we were discussing evolution. And so we kind of did a real quick introduction to it last time. That's fine because for some reason, okay, it is working. Sorry, it's YouTube is really weird, like how it streams. Like you make an event and then sometimes it just doesn't work, and there's just, it's working now. I'm just not going to touch it. So I'm like checking everything on my second monitor. I said 3 things you need to run a stream is a good microphone, a good computer, and water. The second thing, or one of the other things you need with it, is 2 monitors. If you don't have 2 monitors, it's just forget it. Looking at some of the things that we covered for evolution, we went through like a real quick introduction. We talked about how Darwin gathered evidence for it and what it is and what it isn't. All right, so it's not, I'm a horse, I want to reach those leaves up high, so I'm going to become a giraffe. It's more of, you have a bunch of horses or a bunch of giraffes, some with short necks, some with longer necks, and then eventually as something happens in the environment, it leads to maybe food on the ground being more scarce. Therefore, those that have a higher or a longer neck are at an advantage now because they can reach those leaves up top. Actually, I do have a script that I follow, believe it or not. I type out my lessons beforehand and then use those as notes to go through. So it's just not me making stuff up. So when we went through, let me pull up this window. Okay. When we went through and we started talking about evolution, there's two ways that it can happen, and that is macroevolution and microevolution. And what these refer to is they will refer to either large scale or small scale. And that large scale or small scale mostly refers to time period, but also just general scale. So most of the time, if you hear macroevolution, you're hearing speciation. All right, you're thinking about, this is going to be a large scale change over a large period of time, and you're going to get a new species most likely out of this, or elimination of another species or a subset of that species. Compare that to macroevolution, where it's very small scale. It's going to affect not many, but it's going to cause populations to have an impact, and it usually occurs over a shorter period of time. So when you look at something like macroevolution, you can think of the birds that were down in Galapagos that Darwin came across. All right, so he has these Darwin has these finches that he comes across. They're all finches, but they're all evolved or on their islands or respective islands based off of what type of food there is to eat. So if there's an abundance of fruit, you have finches that can eat fruit. If there's abundance of insects, they eat insects, and so on and so forth. That kind of makes sense. The macroevolution does kind of make sense. Microevolution is a little bit more difficult to wrap your head around because it's on a shorter time scale, and it usually doesn't involve this kind of abrupt change in a species. What it does is it will eliminate smaller individuals. And an example of microevolution is seen in E. coli in a lab. If E. coli in a lab are exposed to, so again, you have to have variation. If there's variation and they are exposed to unlimited quantities of a medium, right? They're given enough that they can survive, and well, more than enough to survive. One of the things that they noticed is that these E. coli will become larger. Now, I know it's like, well, that's just them building up body mass, right? It's kind of like how if people are exposed to more food, they'll build up body mass as well. But this is more of when you take those larger cells and then you plate them, one of the things that you notice is that they all grow to that same size, even if you restrict food. So even if you're restricting the amount of things that they can do. Now that's very fast scale microevolution. Usually with microevolution, we're talking about, we're usually talking around like a couple thousand years. So kind of back at this topic, remember in biology, or in science in general, we require one big thing over everything else, and that is evidence. All right. If you make a claim, you have to back it up with evidence. I had a professor one time that says he was a political science professor, and he once said, you are not entitled to your own opinion. You're only entitled to what you can defend. All right. And he broke that down into everything. Because I remember the first thing that he said, he's like, what is your favorite color? And I remember I responded like, my favorite color is green. He's like, why? Their typical response, I don't know. I don't know why I like that color. And he's just like, so then your favorite color is not green. Why is your favorite color not blue? And it's just like, well, I don't know. And I remember he's like, you know what my favorite color is? My favorite color is brown. He's like, you know why brown is my favorite color? Because it just has this warmth and this texture to it that when you look at it, it kind of provides this feeling of security that you don't get with other colors. And that's why brown is my favorite color. But you don't know why you like green. You just like it. And so that was the thing, is that he always said that you need to be able to defend every single thing that you do. And that is the case with science. You cannot make a claim in science without having a way to measure it and then provide those measurements or an analysis of those measurements. And we talked about in class, should you disclose all of your data? And the answer was no, you shouldn't disclose all of your data. You should disclose your analysis about that data. Like, no one wants to see the raw data. Everybody wants to see your analysis of that data. And that's why there's a big portion of people who go for scientific majors in order to do analytics. There's a thing where it's like, oh, you can spin statistics to say whatever you want. That is a bad person doing stats. All right. You should not be able to spin stats any way that you want. And that's why there's rigorous guidelines as far as how to do stats, where you look at certain things. You look at this and you analyze it a certain way. Usually you have your ideas laid out beforehand. For the most part, if someone says you can spin stats to say whatever you want, they have a very surface level grasp of what statistics is. So statistics is a tool that you can use in order to try to analyze things. So we use evidence in order to support our claim. All right. And it could be a claim. It could be like an objective. So looking at previous studies that I've done, where it's like the objective of my study is to find out, we'll use one of them for my master's. Is soil transplant a viable method to control invasive species? And then your answer is, your answer is based off of the evidence that you collect. Now, why do I say evidence and not proof? Well, because we can get into the philosophy of science, but really there's no proof statements. Proof statements are available in math because you could prove that 1 + 1 = 2. I did not prove that soil transplant is a viable method in order to control invasive species. What I did was I provided supporting evidence. Now, a follow-up study on that, which isn't published yet, has shown that as you that as time goes on, invasive species will begin to recolonize. So it was a temporary fix. And again, what does that mean? That means that as we go through time, more evidence is being seen. So one of the common criticisms against evolution is what's called the missing link. All right, the missing link. So the missing link, that is a terrible N. The missing link is if you go from and this applies mostly to humans, all right? But I'm going to use bacteria because I don't want to have to draw out apes and things like that. So you have this bacteria, and then you have this bacteria that has these little hairs hanging off of them. All right, it looks like an eye, but I'll fill it in. And then you have a bacteria that has those same flagellum on, the same cilia on them, but now it's developed a flagellum. And then again, that's supposed to be filled in. And then you get to your final one where it just has a flagellum. So let's say we get rid of this dude right here, right? There is you no longer have any evidence linking the two of these. There's this missing, unknown species that's in this space. So what do you do about that? Well, you know that it should be there. And so with the missing link, finding the steps from here's one, and then it goes to another, and then there's a gap, and then maybe another gap, and then that leads to humans. This space right here and this space right here are referred to as missing links because they are missing links in this chain of making your way from everything that's considered to be pre-human to humans. All right. And he's sad because he can't go outside and he has to listen to me ramble on. And he has a weird shaped head. So this is what we would consider to be or missing links. Now, do we have, the thing is that there are missing links that exist. I'm not going to be one of those people that deny it. But the thing is that as time goes on and we develop, I guess, better strategies for archaeological techniques and things like that, we have found these missing links. And we're starting to find more and more. And I think that this is a big misconception, is that we being scientists have all the answers, and we really don't. And that's the thing, is that it's okay in science to say, I don't have all the answers. It's not okay to say, well, I don't know where this is, so this is probably where it's at, making a claim not based off of any actual evidence. If I said, okay, well, this species right here, this missing question mark species that should be in this gap, we don't know where it is, but judging off of what happened previously with this guy that was missing and we found him, then the best place to search would be in this area, and this is probably where we would start to find those fossils at. So that's sort of the thinking behind evolutionary biology, is that you don't have all the answers, but instead you're going out and looking for them. So what evidence do we actually have? What do we have to kind of support everything? Well, you have to think that when Darwin started off back in the day, right, which is the mid to late 1800s. So you have Darwin. And I mentioned last time that he was a naturalist. And naturalists still exist. I don't want to make them seem like they're not around anymore, but they used to be really common before kind of the 60s, 70s, 80s time frame when genetic evidence became a thing. So we're looking back on this, and what you would do is you would have this animal right here who's happy. And then you have a couple more, right? And then you have one that's just slightly different. One brush. You have one that's just slightly different. Let's say he's a little bit taller. All right, not much, but there is a little difference. Is he still considered to be human? And again, this is usually whenever I do this topic, we're in class, so we can have this discussion. If let's say you're some advanced alien species, right? And you visit Earth in, I don't know, 10,000 years. Let's say 10,000 years from now, you decide to hang out with Earth. All the humans are gone. The planet is basically pretty much barren. And you're going through a cave and you find Shaq, and he's dead right next to a pygmy. Are they both going to be considered humans? Well, I mean, yeah, they are. But looking at naturalists, you are judging all of this based off of the physical appearance of whatever that animal is. And so that is where you kind of run into these questions of, you know, what makes a certain species a certain species. And I know this sounds really dumb, but this is something that naturalists fight through all the time, is comparative anatomy. So you could think of naturalists, they do what's called comparative anatomy. And that is where you take the anatomical or the physical features of an animal, and you compare that to other animals. All right. And the first place that this starts is with embryos. All right. And believe it or not, I wanted to be an embryologist. I wanted to study embryos. That lasted about I'd say it lasted about 2 weeks, and then I kind of got over that. But I really wanted to do embryology, and you look at embryos. So as animals are developing, you have an embryo, which is a what an animal looks like as it's developing. Like, let me find like this. This is an elephant embryo. It looks like a lot of other embryos, and eventually it will develop into an actual elephant as time goes on. But that's what embryologists do. They look at how embryos develop and what they look like through their development cycles. The first thing that they look at is the number of cells that they start with when the first opening occurs, and that first opening eventually becomes the digestive system. And so the two that are the only two that exist are deuterostome and protostome. And deuterostome and protostome is determined about whether either which end of the digestive tract forms first. So with and usually, like, a lot of times when you see these listed, a wind just blew by my window and it made my window shift in the frame. So if you look at so protostomes are where the mouth forms first, and deuterostomes are where the anus forms first, and then they kind of fill in the gaps as they go. I'm sorry, I was trying to look up something, so I was a little distracted, and then the wind. Yeah. So what you're looking at a lot of times are what are called, and this is going back to comparative anatomy. All right, so we're going to go and we're going to look at comparative. Clicked on that. We're going to look at comparative anatomy. All right. So when we look at comparative anatomy, we are looking at what are called homologous features. I don't know why I'm really struggling to draw those. I am not great with a drawing with a mouse. So homologous features, and these are features that are the same. So I actually have a picture. So this is the arm of a human, dog, bird, and then the fin of a whale. And what they have highlighted here are the ulna and radius in the forearm, and then the carpals in the wrist. All right. So as you could see, a lot of these are very similar. I mean, looking at a bird wing, you see that a lot of these, the metacarpals and phalanges are fused, but you see the placement of where these different bones are. Now, do we fly? No, but a bird also doesn't use its wings to manipulate stuff like what we do. It doesn't turn its wing in a similar fashion to how we can turn our forearms. Same thing with a dog. A dog doesn't necessarily pick stuff up and hold it with its hands or use its hands in a similar function. Same thing with whales. They don't use their flippers are more for movement and balance, compared to with us, our arms are for manipulation. Grabbing something and moving that, or holding it, any other thing, grasping, attack and defense for fight or flight, things like that, balance. So we use our arms differently than other animals do, but you could see the composition is very similar. In a human, you have your humerus, ulna radius, carpals, that order. In a whale, same exact thing in that order. All right. So you have those same exact things in those in that order. And this doesn't necessarily just apply to features that are visible like this. Another example that I have is feet on a snake. Yes, some snakes have feet. Do they use them to walk? No, but they're there. We have a tailbone. All right. Whales actually have hind leg bones inside of their bodies. So why do these exist? Well, these are leftovers from when the species evolved, and it just never fully got rid of them. It's like our tailbone. Our tailbone served a function at one point, but as being humans, we no longer have a function for our tailbone. Although we did say as biology teachers one time, we were talking about if you can have an extra thing on your body, what would it be? And the one that we kind of came to the conclusion was a prehensile tail. So basically a tail that you can control. Like that would be so useful for just doing all sorts of stuff. But that's a side conversation. But if you look at this snake has legs. These are leftovers, all right? But they are important when you refer to evolutionary ecology or evolutionary biology. Why? Because this gives you a level of comparison. What other animals exist out there that have tails or tail bones, but no external tail? Well, if you look, a lot of apes do. And we share a lot of similar features to other apes. So if you look at an orangutan, chimpanzees, things like that, the way that the hands work, the facial structure, a lot of these things are in common. And those are those shared traits that naturalists use in order to do comparative anatomy. They look for these homologous features or similar features, all right? And because of that, scientists were able to build kind of this evolutionary tree based off of what was currently visible at the time. And it kind of makes this odd point because you have this old way of thinking, right? Where we have to look at animals and see how they compare to other animals. And then what happens when genetic evidence comes around? Well, now you have another way to test for this. And is any one way right or wrong? Well, with genetics, it mostly depends on how you use it. But for the most part, no, not really. Neither way is wrong as long as it's used correctly. You know, it's sort of that whole thing about like I hired a video editor. So the thing with that is, do I care what program he uses? No, he can use Adobe Premiere. He can use, Light Studio, DaVinci, Vegas Pro. Doesn't matter. All I care about is the end result. And that's sort of the same thing with some of the techniques in evolutionary biology to an extent. You do want to know what technique was used so that you can go back and check, because that is one thing, is that every single thing in science has to be repeatable. That's why we, excuse me, that's why we look at things like how we spell our methods out. This is exactly what we use. This is exactly what we did. Like, it's to the point where I put down that I use ArcGIS and then what version in order to do my spatial analysis, because between different versions there may be like the math might be done differently on the back end of it, or there might be an algorithm or a shortcut that's used that's in one version but not the other. Okay. So I hope that kind of clears up some things with homologous features. the next thing that you have, it's a lot more rare. Do this in orange. It's called analogous. I did click orange. It's called analogous features. And this is a little bit more rare. So homologous features are features that are shared by organisms of kind of the same family. But if you look at analogous features are features that are shared by distant relatives. And a lot of this analogous features sometimes involves that they are features that were produced in isolation. So they were produced in isolation. And that doesn't mean like how right now we're in isolation. What it means is, so an example is an Arctic fox and a bird. There's some birds that go through seasonal changes, changing their coat color from darker to snowy white. Now, yes, everything has a common ancestor if you go back far enough, but they don't have a recent common ancestor. So it's likely that this trait in birds and Arctic foxes have evolved separately from each other. All right. So they have evolved separately from each other. And again, this tends to be more rare, but it is something that you have to talk about because you need to address like, okay, well, what does this mean? Because again, if you're presented with a problem, try to find an answer to it. So what does this actually mean? All right. So you have these two species that they can darken their coat color in the summertime, and then as the winter approaches, it becomes snowy white. Let me show a picture of that. So here's an example of it. So you can see that this Arctic fox has a white coat, and then this bird also does too. They're both in snow, but in the summertime they'll darken up. And so what this has done is this. Now, does this have two separate features? Yes. Does this bird really need to hide from or need to be stealthy? Well, yeah, because it has to worry about being eaten. Does this fox have to worry about being stealthy? Well, yeah, because it has to find food. And remember, animals have to do three things. They have to find food, or they have to find a mate, find food, and not become food themselves. This fox doesn't really have to worry a whole lot about not becoming food. All right. Yes, there are things that hunt it, but they're few and far between. For the most part, it has to worry about finding food. So this white coat color allows it to sneak up on other animals, while this bird has to worry about becoming food itself. So it has to be able to blend into its environment. So that's where you end up with these two separate cases. So that's my last picture. You have this bird. Which mice stand out? This is something that it's kind of awkward asking myself questions, knowing that I'm going to give the answer to it. But this bird flies overhead. Which mice is it going to select against? Well, it's going to select against the ones that stick out. And then after a while, these mice are going to reproduce. Remember, it has to find a mate, find food, not become food itself. These mice have succeeded in not becoming food themselves. They've succeeded in finding a mate, and they found enough food that they can now reproduce and make it to the next generation. Notice how in this you have, 6 light-colored mice and 3 dark-colored mice. But as the end result, now you end up with 6 light-colored or dark-colored mice and 2 light-colored mice because of this shift that has happened. All right. So that will kind of finish up. Talking about naturalists. Now we're going to get into molecular biology. So molecular ecology is just what it sounds. Ecology is how do things interact with their environment. Molecular deals with genetics. How do genes interact with their environment? How does an animal's genetics interact with its environment? All right. And so this is where it becomes a lot more complex. I actually took an entire class in college called molecular ecology. You literally spend your entire time in that class only talking about the genetics of, okay, this animal looks like this and this other animal looks like this. What does that say about its genetics? All right. And what this does is this looks at, yes, it looks at cell functions. All right. But what it mostly looks at is with these cell functions, what are the proteins? All right, what are the proteins and how are they built? I'm just going to put built because that's easier. All right. So what are the proteins and how are those proteins built? So an example. This house, all right? We know it's a house because of how it looks, all right? We know that's a house because of how it looks. This is also a house because of how it looks. Yes, this house has a pool and a massive garage and a okay sized yard and grass everywhere where there's outdoor spots and a helicopter landing pad and multiple places to hang out, and it's made out of glass and concrete the whole way around. But it's still a house. Is this thing really worth $46 million? And it's a YouTube video. I got on a kick one. This is kind of off topic, but I got on a kick one time of watching like real estate, because real estate agents are starting to post videos online of like high-end houses, especially the real estate agents that deal in places like Beverly Hills and stuff like that. But they're posting a lot of videos with like, this is what you get with this house. That way you can go back and review it and see it, and it's not necessarily spending a lot of time. And like this house in Palm Beach, $43 million, is still a house because of the features that it has. And it looks different from this one. Even though they're pretty close in price, it looks different, all right? This one's a little bit more modest as a $43 million house can be. And this one's a little bit more extravagant, modern, while this has a classical design. And this is your average house. But they're still the exact same thing. They're still a house. And so it's how you build that house, all right? So the protein being the house and how it's built being what we're measuring in molecular ecology. So let's use cytochrome B, all right? Cytochrome B is a protein that everything has. It's used in the electron transport chain, all right? It's used in the electron transport chain, and everything has this, but how it builds it is slightly different, all right? So let's look at this versus this. One difference, all right? One difference. And depending on where it's at and how this letter falls and all of that stuff, it might result in no change in protein. This region might not even code for a protein. It might be a kind of a blank region, and we sometimes call those microsatellites. Even though I do satellite with amateur radio, I struggle with spelling the word satellite. Okay, so we have these microsatellites, which are uncoded regions, or we can use coded regions. So how do things build these? Well, there is a website that you can use and it's run by the National Institute of Health, or the NIH. And what they do is every single time people code stuff, they usually submit it to NIH, and then that gets stored in this genetic database, literally the raw code. And so then you can go back and do comparative anatomy using a program called Mega64. And I know there's a YouTube channel called Mega64. So if you look it up, you have to do Mega64 program. Mega64 genetics program. And you can download this from a company called Mega, and this is it right here. And what you do is you can actually leverage information from the NIH database. So you could type in humans, chimpanzees, all of this other stuff, and it'll build a phylogenetic tree and show you the frequency of how often letters show up within that. And so this is the information that molecular ecologists use in order to determine, okay, where does this species fall at in the evolutionary chain compared to its other relatives? At least that's what has been done previously. So what is being done currently? Currently, what a lot of molecular ecologists are doing is they are looking at, let's say you have this landscape, right? This is a landscape, and you have, say this is a mountain range that separates two populations. So you have population 1 and you have population 2. Let's say these are populations of, I don't know, let's say they're mice, right? That makes it easy. They're mice. How do you measure the difference between these over time? All right, let's say that this mountain range popped up 5,000 years ago. So recent, right? So this is a recent geological phenomenon that has occurred, that this mountain range is now separating these two species of mice. They're still the same species of mice, but how do you measure the difference between them? Well, one of the things that you can do is you can look at microsatellites, these regions that don't make anything. And because they don't make anything, there's no control over it. If you mess up cytochrome B, if you mess it up, you won't live. If you mess up a microsatellite, whatever, it doesn't do anything, right? So imagine it as you're in your car, right? You're in or you go out to your car. There's a hole in, let's say there's a hole in your seat. Does that affect the function of the car? Well, no, not really. Your car still functions. But what if there's a hole in your tire? Well, that's going to be pretty detrimental to the performance of your vehicle. So cytochrome B is a tire. It's required. Microsatellites are seats. If they take a little bit of damage, no big deal. So what you do is you look at some microsatellites in here, and you're like, okay, how different is population 1 from population 2? And so you look at the microsatellites, you kind of code through everything, and what you're looking for is, let's give it a name. All right, let's give it a name. So again, this is kind of awkward because I teach this, like this makes more sense one-on-one. So if in this example, we had homologous features. In this example, we have homologous genes. And these are genetics that are shared between populations or species. All right. Now, between populations, it's going to be more difficult because remember, there's no reason to fix what isn't broken. So there's no reason for cytochrome B to change, but the microsatellites might change a little bit. But let's compare cytochrome B in a human, or let's compare insulin in humans and chimpanzees. So insulin, which is a protein used to regulate sugar control or blood sugar levels. In humans and chimpanzees, they're 98% similar. right? Which means comparing all of these letters, they're going to, out of 100 letters, 2% is going to be wrong. And when you look at proteins, for the most part, when it says 98%, what it means is out of all the thousands of, or not 2% will be wrong, 2% will be different. But looking at the proteins, out of all of the thousands of amino acids that build this molecule, 2% is going to be different. All right. So why does that matter? Does that mean that we're 98% in common with the chimpanzee? Well, remember, you're using this as a comparative feature. So then let's look at humans and chickens. All right. Humans and chickens have about 64% that's identical between our how we build our insulin, which means that you can make the assumption that humans and chimpanzees are more closely related than humans and chickens. And I know I just named one protein. All right, I just named one protein in 3 animals. But what you do is then you start to look at more things. You look at cytochrome B, you look at other proteins that are constructed, and then you start to develop a bigger picture. If you just look at insulin, you're painting with blue. If you look at insulin and cytochrome B, you're painting with blue and green. If you start to add on more and more and more, then you start to add more colors to your color palette. All right. And that starts to paint a better picture for you. So you're really adding to it by looking at homologous genes. Now, is it possible for a scientist to look at every gene between 2 animals? Given enough time? No. I mean, if you live forever, you probably could, as long as you had access to a constant amount of those. But for the most part, no. So this goes back to the collaborative nature of science. All right. And I know, like, we have emphasized a lot on the 7 C's and things like that, but really science is very, very collaborative. It doesn't do any good if you find out information and then you keep that information to yourself. All right. You need to share that information. You need to share that information out. And because of that, we have a collaborative pool of information. I'm going to switch over to this and then I'm going to close all my paint windows. So you have this collaborative pool of information. And looking at that pool, you really do kind of add to it, right? When I did my research, I don't think I have a copy of, I don't have any of my research on my computer right now, but if you look at a lot of what I did for my research in the introduction, a lot of it is citing what other people have did or other researchers have done in the past. So, when they say one of those things like stand on the shoulders of giants, like I could not have done my research without other people in the past. There were these 2 scientists at, I want to say it was University of Cincinnati, but I could be wrong about that. And they, one of them was the professor and the other one was the PhD student, and they were doing research on calorie pair. And I probably cited like at least 3 or 4 of their articles the whole time I did research. And so it added to that collaborative nature of science. Have I ever met them? No. I've never met them. I actually had a chance to meet one of them. She was presenting at SER, Society for Ecological, this is a mother lode for the name. It was at the Midwest annual meeting of the Midwest Great Lakes chapter of the Society for Ecological Restoration. And it was in Bloomington at IU. And I was presenting information over how I was trying to use fire ecology to control calorie pair, because at the time we were still waiting on last minute data to come in for another research project I was doing. So I decided to present calorie pair. And I had presented Calorie Pair the year before, so it was kind of a follow-up with like, hey, if you were here last year, you saw this. This is 1 year later. Here's all of the data that we've collected in that time frame. And so apparently she was there, but she had left to go some other place or something like that. So I didn't actually get to meet the person that was doing it. It was two ladies that were doing the research, and the professor or the advisor was there. And I never got a chance to meet them. But all of the research that I did, a lot of it was based off of information that they had collected previously. And because of that, we added to this pool of Calorie Pair information. So when you're looking at, okay, how do we know that humans and chimpanzees are closely related? Because you had some researcher out there that was like, one day he was like, you know what? I want to look at doing comparative genetics, and I want to look at how is insulin made in humans versus chimpanzees. I'm going to look at genetic amino acid composition, whatever it is. I'm looking at this, and this is how I'm going to base my research, or I'm going to base a paper off of this. And then once he gets that information, he shares it with everybody. So now that information is out there and it's available. So looking at that from another standpoint of, okay, now we have this one bit of information. Some other researcher is going to go out there and look at, all right, I want to know about cytochrome B, because cytochrome B is the easiest. Everyone does cytochrome B. If you take molecular ecology, if you take any sort of genetics course that deals with evolutionary ecology, you're going to talk about cytochrome B. If you take an evolutionary ecology, or if you take a genetics course that doesn't have anything to do with evolution, you're still going to end up talking about cytochrome B. It happens. It's kind of like fruit flies. It's just a part of the experience. And so you look at another one, rhopsin, a protein that's found in the eye. So you could use those as comparison. Now, the thing is, do we look at insulin in plants? Well, no, not necessarily. So there are certain things that you are looking at, certain genes or certain traits or whatever, right? These are these facets that you look at within different animals, and you use those to compare, and it adds to that larger pool of information. So that's why I said there's no such thing as proof. What you're doing is you've constructed a bullseye, and you know that through practice, by throwing your dart, you're going to get better and better and better and better. So you're going to start to get closer to the bullseye. And that's what the goal of science is that we're taking all of this information, and that's allowing us to get closer and closer to the bullseye. Will we have all of the information? Probably not. But we're cool with that. I know that in my lifetime that there's answers that I'm not going to get, and problems that I'm never going to solve, right? I'm never going to know everything there is to know about Calorie Pair, even if I tried. And so you kind of have to come to that realization that, okay, then what's my goal? Why do I do this research? Well, it's to contribute to other people, because out there, someone else is going to be contributing their stuff, and it's just going to pile up into this collective pool of knowledge. All right. And I hope that kind of makes sense. I know that was kind of a little rant at the end. The last thing that I wanted to do was, it's been a while since I've done a cultural recommendation. Cultural recommendation being that students now have quite a bit more time off to do whatever, right? And I know that every single person tunes in and watches these videos, and so when you're not watching these videos, or you're not doing homework for other classes or anything like that, then what should you check out in order to kind of make the most of your time, since we're kind of stuck indoors? I saw on Reddit this thing. It was like, try to work on improving, try to work on either learning a skill or improving at another skill. So one of the things that I'm doing is I'm trying to learn Morse code. Why? Because I'm a nerd. Why not? But I was really interested in, excuse me, I was really interested in cultural anthropology, mostly in folklore, from a cultural anthropology standpoint. How do certain things end up the way that they are. And there was a cultural anthropologist, last name was Childs, and he came up with these things called Child's ballads. It's sort of like how if you look at Twinkle Twinkle Little Star and Happy Birthday, the melodies to it kind of go the same. So there's this website. Let me go to it. It is called 100 Years of Rock Visualized in Less Than a Minute. So it starts off at the very top, and then it kind of evolves through this time frame and shows you how different versions came together and different versions kind of merged and or split apart and things like that. And yes, this is rock, and this is at concerthotels.com/100-years-of-rock. And so you end up with this sort of evolutionary tree of where rock music came from. So you start off kind of pre-1900s. It's a lot of folk songs, right? So gospels and hymns are folk songs. Work songs are songs that were sung either for slaves in fields. You also hear it quite a bit like in on ships where you have to pull a line. You're trying to pull something heavy. One individual can't do it. So how do you know when to pull at the same time? Well, you sing a song that has a rhythm, and every other word or every other part to it, you are reaching forward, pulling back, reaching forward, pulling back, and you're doing it very methodical so that and how do you keep everyone on pace? Well, you sing a song that goes with it. When I was in the military, again, when you're running on you tend to sing cadence. A lot of it is to get you to if you're singing while running, you're breathing in deep, you're opening your lungs up and things like that. You can perform a little bit better, but a lot of it is to keep everyone in pace. The reason that your left foot always hits exactly the same and your right foot hits exactly the same along with everybody else in the group is based off of these songs. Spiritual songs being the same thing. These are songs that are separate from hymns and gospels, and then kind of how they evolve into these different things. So you can see that like blues music has these blue arrows, right? It makes sense. That ends up as boogie woogie, which eventually comes down and becomes electric blues and then jump blues. And then there's a slight change where you get R&B, and then that becomes rock and roll. And then there's all sorts of mergers and changes as you go through this. And one of the things that you can do is you can click on one of these and it will give you an example. So we'll click on rock and roll, and it pulls up Rocket 88 by Jackie Brenston and his Delta Cats, which was the first ever rock and roll song. And it gives you the option to download it on iTunes, but you can use Spotify or Tidal. But this is a really cool way to see how evolutionary biology has been applied in other fields. And I don't know, this is why I became really interested in folklore, is because you can actually use this to trace the origins of where music or music genres came from, where they emerged at, and what their influences are. So that's something that if you're interested, you could check this out. This website's really cool. It'll teach you a lot about where certain songs came from, but also just to open up like earlier today when I was looking at this, I clicked on German electronic metal, which is something that I would never probably click on my own. And like, what is mathcore? And you could click on these different things and get some really cool ideas for music. So that is my cultural recommendation. But thanks everyone. I'm going to be jumping over to Google Meets after this. The link will be posted in PowerSchool. I am getting your guys' emails. I am putting in your guys' assignments. So keep those coming. A new assignment has been posted. We're supposed to be posting 3 assignments per week. This assignment is your 3 assignments for the week. It's pretty big, which means don't sit down and try to grind through it all at one time. All right, split it up. You're not going to have all the information right up front. We'll, again, on Wednesday when we talk about biodiversity in with geographical and fossil records, we'll be adding a little bit more to that, so you'll be able to complete the assignment a little bit better. But keep doing or do those assignments. Don't do that assignment all at once. Spread it out over a few days. You should be able to have it finished though by Friday if you work on it for about 30 to 40 minutes every day. And then remember to tell everybody that these videos are out there. If they have questions about anything, send me an email. Check PowerSchool every few days. But that is all I have for today. So I will be over on Google Meets in a few minutes. I'm going to use the bathroom, but I will post that link into PowerSchool. But thanks everyone for watching.